¿Las paso o tú las controlas? Es que no las tenemos.
Cambió así.
Lo voy a estar presentando yo.
Hola, mucho gusto.
Good morning, everyone, and welcome to the first part of this assembly. For this morning, we have a talk title From Brain Development to Slug Regeneration by Dr. Alfredo Varela. Dr. Varela completed his undergraduate studies in biology at the Autonomous University of Nuevo León, and his doctoral studies in molecular genetics and microbiology at the University of Medicine and Dentistry from New Jersey, USA, specializing in mutational studies in system retrovirals. Subsequently, he developed postdoctoral work in the field of neurobiology of the development at Guy's Hospital at the University of London, today part of King's College. Dr. Varela is currently titular investigator C of the Institute of Neurobiology of the UNAM. He belongs to Pride UNAM at level C and the National System of Researchers at level 3. Dr. Varela's research work focuses on the study of the molecular control of embryonic development of the vertebrate nervous system, using embryology methods experimental with transgenic birds and mice, molecular biology, and genomic approaches. Please give him a round of applause.
Good morning, everyone. First, I would like to thank the organizers, especially Professor Akpasama team, for the invitation to be here with you, and I want to thank all of you for being here. It is a great honor to be the first presentation at this assembly. And it is great to see the campus. I was here at the opening ceremony a few years back, and it's great to see how it has changed, and it's nicer and nicer every time. And it is great to be here. Today I'm going to talk to you about some of the research we have done in my laboratory over the number of years. And I'm going to tell you how, from studying brain development, as my title indicates from studying brain development, we are transitioning into studying regeneration in slugs, in common garden slugs. This transition might seem a bit drastic, but you will see that the things that we're interested in is how changes in shape during development are regulated by genes, by gene networks, by gene regulatory network, as we call them. So this transition, even though again might seem drastic, it is within the same field of developmental biology, which is what I'm interested in. We are interested in studying how changes in shape, in changes in the evolution of the tissue along development is regulated. And that is the basic principle that we will follow today. Beginning with the brain, I'm going to tell you about some of the experiments we have done addressing from molecular aspects to integral view of the whole brain, how the whole brain develops in different models. So to begin with, in the first slide, we can see what I'm trying to show here is that there are many different shapes, sizes, colors, and habitats and behaviors of vertebrates. And you can see some of them here. But despite this great diversity, all of them have brains that derive from very similar embryonic structures. And in the next slide, we see that these are diagrams, cartoons of the brains of many different vertebrates. We will not see all of them, but I just want to mention some of them. If we see to the left of the slide, we can see that there are different groups of fish, different types of fish. And then on top, in the center, we can see amphibians. And to the right on top, mammals, and then birds and reptiles at the bottom on the right. And the colors of the different brain regions indicate regions that are equivalent. For example, if we look at the dark green, that indicates a part of the nervous system or the brain that is involved in olfaction, that is the olfactory bulb. And then if we look at the lighter green, that is that structure that in humans is the cerebral hemispheres, and that's the cerebral cortex is located. And so you see that light green is present in all of them, despite the different shapes and sizes of these structures. And then we look at the orange structure, that is the cerebellum, and then the yellow bit, the yellow part is the brainstem. Most of it is what we call the hindbrain, the posterior most part of the brain. So again, despite all the morphological differences in vertebrates, and then all the differences, even at this stage, these are the structures of diagrams of the adult brains of all these animals. And we can see that they all have the same structure. So they derive from the same embryonic structure, embryonic components. So if we study, let's say, the development of the chick brain or the mouse brain, we can learn many things that take place even in the human brain. So that's how we can understand more about how the human brain develops by studying other species that are model organisms that are amenable to experimentation in the laboratory. In the next slide, we can see, if we could turn these slides off, just in the front, just a bit. Yeah, that's not that great. There are many different types of neurons in our brain, in an adult brain. I'm showing here just one type of neuron. These are the pyramidal neurons of the cerebral cortex. These, I mean, the whole image on the screen would be a couple of millimeters of the brain cortex, in the equivalent to the brain cortex in humans. This is an image from a mouse, and each neuron has been changed genetically so that it expresses a combination of fluorescent tracers, what we call reporter molecules, fluorescent tracers. In each combination of tracers gives it a specific brain, a specific color, a specific shade or intensity of color. So these are pyramidal neurons in the cortex, but there are many different types of neurons in the brain. There are even some that we don't know about. I mean, there are colleagues at our institute that are studying the structure of the adult brain and discovering new types of neurons, different shapes, different forms, different functions, perhaps. And many of them, we don't know the function that they are involved in. So in the next slide, we can see that this will be like a stereotypical neuron. Perhaps you have seen this in some of your classes, some of your lectures. This would be equivalent to a motor neuron. You can see on the left-hand side the cell body that contains these spikes, these projections that are the dendrites. And then a continuation of this cell body going to the right of this slide, the axon, you can see the axon. That is a cylinder that is a continuation from the neuron. And then at the end, we have the axon terminal. And so the axon is the structure that conducts the electrical impulses that allow a communication between neurons or between neurons and other organs, for example, the muscle. And if this neuron were involved in forming the neuromuscular junction, for example. And the dendrites, that's where most of the signals that the neuron receives arrive to the neuron. So the signals arrive to the dendrites, and the signal is conducted along the axon, and then goes all the way to the axon terminal. So we done some studies to understand how these neurons form and how do they establish connections from the origin where the cell body is located, all the way to the end, where the axon terminal ends, and how it identifies the target it will connect to forming functional circuits. So in the next slide, we see, this is just to illustrate, to remind me to tell you that brain connections are made out of axons, the axons of the neurons. Some neurons will project long distances in the body. There are some neurons that are located in the brain, they project all the way to the spinal cord. And the longest neurons, the longest known axons will be those in animals like a giraffe, where you have a neuron in the central nervous system, in the spinal cord, and then you get the axon terminal projected all the way to the legs of one of the legs of the animal, making them perhaps the longest projecting neurons in nature. In the next slide shows something similar, but this is to show that that the brain is connected. Different structure, different regions in the brain are connected in different ways. Some of them are very short connections because there are some neurons that project very short distances from where they are located, where they are born, or where they end up in the brain. And some of them project long distances, as I mentioned before. On the next slide, we see an example of how we have how we study different aspects of the developing brain. In this case, what I want to illustrate with this image is that we use transgenic animals. We use mice and mouse embryos because, again, as I mentioned before, if we study how the brain develops in a mouse, we can learn many things about how it takes place. The same process takes place in the human brain. But in this case, we are studying the consequences of the absence of a single gene that is involved in the development of different structures in the brain and in other parts outside the brain as well. What we can see here is that the absence of a single gene causes dramatic alterations in the brain. We can see on the left-hand side, the column of images on the left-hand side of this slide. This is the telencephalon. This is the part that in humans gives rise to the cerebral hemisphere. And at embryonic stages, it's just like a balloon, a tiny structure. A balloon has got a wall, a cellular, a wall made out of cells, a very complex structure involving cells. And in this balloon, this telencephalon, which is the part that is the anterior-most part of the collecting brain, there are different types of neurons. We have the early stages of This is the one type of neurons develops. These are the Cajal red cells, and you can see them staying by a method that is called in situ administration, a method that detects the messenger RNA, that is the product of the expression of this gene. We see the messenger RNA that is staying in dark, brownish, purplish color. And on the left-hand side, you can on A, you see the lateral view of the telencephalon. That is, if the brain is like this, like me, so it will be a view like from this side. And on C is the same preparation, the same structure, but it's seen from the medial side. That is, if we separate the balloon and we look at it from the medial side, and we can see that in both sides we have those neurons, the Cajal red cells. And these neurons, one of the functions of these neurons is that they have an organizational function because they are required for the formation of the layered structure of the cerebral cortex. In the absence of these neurons, the layers of the cortex do not form, and there are many consequences, behavioral and anatomical, physiological consequences to this. So that is one stage. The E11.5 is the embryonic stage 11.5 after this 11.5 days after fertilization. And then at the bottom we see a mouse that is these are controlled dish, normal from normal embryos. It's 1 day later, you see that the number of cells has increased. But if we look at the right-hand side, the column of images is showing the same structures, staying the same neurons, and we can see that most of them have red cells are absent. And this illustrates how well, several things. One, how dramatic the consequences can be of the absence of a single gene, and how powerful these methods are that allow us to detect and study these neural attacks. And like experiments like this, there are many groups around the world studying this type of thing, analyzing the function of hundreds and thousands of genes in a directed way. We are going to make a reference to this later when we address or when I describe how we study all the genes that are expressed in a similar structure in mice and in chicks. But this is looking at a single gene and looking it's like a very surgical approach. Yeah. So we inactivate one gene and we see the consequences. And then on the next slide, we can see a different way of looking at things, a different way of studying things. In the previous one, I was looking at the consequences of the absence of a single gene. Here we are now looking at more okay. So here we're looking again at a lateral view. This is a lateral view of a brain, but here we have stayed with a specific method that is called immunohistochemistry, immunostaining with fluorescent tracer called fluorescent molecules. All the axons, in this case, we are staining all the axons that are already there. I mentioned that neurons have a cell body, an axon, and then a terminal. But in earlier stages, they begin to form those structures. In those, the axon grows along different pathways, and this is what it's showing us here. And that's one of the things we have studied, how a neuron that is formed in one place projects its axon along a developing environment that is changing, and how it finds its target and makes a connection. But in this case, we were looking at, actually, we were not looking at cell migration, but we discovered a new neuronal type that nobody had seen, and there's a great deal when one sees that and when one talks about this, because this is something that no one had seen before, and it's just fun experiencing that. So this is a lateral view of a developing brain of a mouse, and then you can see here, this will be the forebrain, the structure that gives rise to the telencephalon, to the cerebral hemisphere that I mentioned before. This here is the midbrain, and this is here the hindbrain. Slug. Yeah. Este se murió. So and we are here on the right hand side, we see a close up of what's going on the midbrain. And here you can see on red the axons and all cell bodies and axons of all neurons. All neurons are already present there. And then on yellow, we see the result of the combination of a dream stain that detects a specific neuronal type. This is we use an antibody that is tyrosine hydroxylase, which detects a specific type of neurons. And what we saw first, we didn't expect to see neurons, that type of neurons at this early stage. And then the second thing we saw after the first excitement of looking at this, at something new, is that all the neurons were attached, opposed to this axon. So that suggested to us that these neurons were migrating along those pathways, along the pathways afforded by the axons that were present there. So we looked at different stages, and then we can label them. We can, let's put a fluorescent dye here that labels all the cells that are present there, and then we can follow them and we can see where they end up. And what we saw is that they migrate caudally, and they go from this region, they migrate all the way here. To this stage, what we published as results, so the references here, but we still don't know what those neurons are. But they have a very interesting behavior. And the point here is, one, to show what we're doing or what we have done, and two, to remind me that there are many different types of neurons that migrate along different routes in the developing brain. So we looked at genes, now we're looking at how neurons are born, and then migrate at some distances during the developing brain. On the next slide, we see something related to that, but now we stain the axons with a different color using the same antibody, but now using a different fluorescent marker. And we can see here, this shows more in more detail how the axons, which are shown here in green, this is a close-up of that image on the left. And we can see that these cells, these migrating cells, are growing along these tracks. And to test, one of the things that we do in science is that when we make observations, we venture an explanation of what is going on what we see. And then that automatically that observation can become a hypothesis to what comes next. So in this case, we say, well, if these axons are if those neurons are interacting with the axons as a way of moving around the brain, in the developing brain. So we put that to a challenge. What happens if we alter somehow remove the axons so that we can observe the migration? So removing the axons makes those neurons get stuck. They lag behind, or they get stuck in the midbrain, and they don't migrate all the way to the hindbrain. And again, now we have more evidence to that hypothesis we have that the neurons require those axons to migrate along the route. So that's how we start building complex models that explain complex behaviors. In this case of neurons and axons. Next one. Thank you. And this shows now we're shifting a bit. We're going into a subject that I mentioned before, but now I'm going to go more in depth a bit on this subject, which is how the axons that are born, that are projecting out of a cell body, a neuron, make their way, find their way to the target, and then they will connect to that target. This is similar to the image that I showed you before of a stereotypical neuron, but in this case, this is a developing neuron. This is from an embryonic brain. we see again the cell body, we see the axon. In this case, this is a growing axon. And at the end of the growing axon, we have something that was discovered by Santiago Ramón y Cajal, a Spanish neuroscientist, who described just by looking at static images, he imagines a dynamic process that was taking place just by because they didn't have all the technology that work now. And it took over 100 years for scientists to confirm this, the observations or the hypothesis that Santiago Ramón y Cajal put forward. So at the end of the axon, the growing axon, we have what is a structure, what he called, Santiago Ramón y Cajal called the growth cone. And he imagined that this growth cone was exploring the territory, detecting signals, and responding in growing or retracting. And that is exactly what happens. It's amazing that this observation work, that this hypothesis was put forward over 100 years ago. So now we're going to address, I'm going to give you some of the evidence we have obtained as to how the neurons grow in the different territories in the developing brain. And one of the things that derived from Cajal's observations was that axons grow towards attractants and grow away from repellents. And that's what I'm going to show you in the next few slides. This is the basic idea. We have a growing axon. This is a representation of the growth cone. And as I mentioned before, the axon is an extension of the cell body of the cell. So the membrane of the cell is continuous from the cell body to the axon to the growth cone. And on the cell membrane of the growth cone, there are receptors, these molecules that will detect the molecules that are in the environment that are attractants or repellents, and they will grow away from a repellent, as is shown here, or towards an attractant, as is represented by the green signal there. So that's the basic idea in that, and then we did some experiment to test that, not in general, but for specific neuronal path. And on the next slide, we see some of those, how we started looking at this. One of the things that we, at least in this field of neuroscience or experimental biology do, is that we simplify the system so that we can make observation. It's very difficult to look at this in a living embryo, in a living animal. So we make a model of that so that in the laboratory, we have a representation of that process so that we can do some experiments, some manipulations, so we can look at the behavior of axons, neurons, etc. And one way of doing that is taking explants, that is taking a bit of the developing brain, put it in culture, and then look at the behavior of axons, neurons, or the whole tissue as well. So we can look at, we can study things that are going on a developing brain. We can look at that on a cell culture dish in the laboratory. And here I'm showing an explant of a brain region that contains dopaminergic neurons. And these neurons are important, as I'm going to show you a bit further in one of those slides, next slides. These neurons are important because the selective death of these neurons is what causes most of the symptoms of Parkinson's disease. These are the dopaminergic neurons that are located in the midbrain, right? In one of the regions that I showed you before. And we can see here, this is a lateral view of a diagram of a brain, rat brain, and we can see two neuronal nuclei. These are dopaminergic neurons here, the A9 and A10. I'm going to concentrate on the A9. So this is a group of neurons called dopaminergic neurons because of the neurotransmitters and neuromodulators that they express. They receive that specific name. And they project, this is a diagram of an adult brain. So they project from that location here on the midbrain, all along they project what we call rostrally to the anterior part, and then connect to this region that is called this striatum. And this connection is very important for motor control. So in the absence of those neurons, many of the symptoms of Parkinson's disease ensue. So how we study this? There are many ways of doing this. And so we started by taking from an embryonic brain, taking this region and studying the behavior of those neurons. And then on the next slide, we see an example of that. So if we take that region of the brain, put it in culture in a 3D matrix that we made with rat collagen. So it's like a drop of collagen, but we put the tissue in the middle, and then we change the pH of the tissue so that the collagen congeals and forms this 3D gel, and then traps the tissue inside. And then out of the tissue, neurons grow. The axons of the neurons grow. And then we stain them with the same antibody that we use for the other studies in which we detect tyrosine hydroxylase, which is a way of identifying dopaminergic neurons. So, and then the basic setup of the experiment is like this. We have an explant in the center, and then on the two corners, as shown here, on top right and bottom left, we put cell aggregates that have been transfected. And cell aggregates to which we have introduced expression vectors to express molecules that are called, that we call axon guidance molecules, molecules that we know control or may affect the growth of axons. In this case, we were testing semaphorin 3A. This is the same three members of the same family, semaphorin. They're called semaphorins because of the analogy to traffic lights, which in Spanish is semáforos. So they're called semaphorins. And what we can see is that the axons, the dopaminergic axons growing out of these explants are attracted by semaphorin 3A, and we can see that they grow to here and grow to the other corner. In the middle, we see that they are also attracted by semaphorin 3C. And then on the right-hand side, we see that they are repelled by semaphorin 3F. So members of this family of molecules, semaphorins, some of them are attractants, some of them are repellents. And this will change if we go to a different neuronal type. For a different type, this will actually, all these molecules were originally described as repellents. And then we found that they can be attractants as well, and other people in those other neuronal types. So now we can see how we can ask specific questions of the specific neuronal types and learn a lot about that. And then perhaps at some point, try to use this information to devise strategies to control the growth of axons if we were to develop a therapeutical approach for Parkinson's disease. But this is analysis or studies on at the basic level. So now here we are going to look at a different thing about the same neurons, right? So again, we begin with a panoramic view. This is again the forebrain, midbrain, hindbrain of this region here. And these dopaminergic neurons that I mentioned before are born in this region here of the midbrain. So we were very busy studying how the axons of these neurons respond to attractants and repellents. But then we do just along these experiments, we realized that by the time these neurons begin to project their axons en route to the target, to the eventual target, we saw that there are many other axons that are present there. So it's not as if they are projecting in a vacuum. There are many other things that are already there, and say, well, maybe they are not just responding to specific guidance molecules. Maybe what if they are using a pre-existing highway that is already there? And the highway will be those axons that are already there, because here at this stage, the neurons have not projected. These dopaminergic neurons that I have shown in red, they have not projected yet. And when they begin projecting, there is a continuity all the way from here to the normal target, to the place where they project to. So we say, well, maybe if we start looking at what controls, if there is some kind of interaction, maybe we might be able to find whether that interaction is relevant for that projection or not. So the first thing is we wanted to see if there is that interaction. So we stained with one antibody all these axons, and then with on green, and then we stained the neurons that are the center of our interest, the dopaminergic neurons, in red. And what we can see here is in different places, but we can see here, for example, here where you see the yellow lines, that means that the green and the red merge and give yellow, right? Just by the superposition of colors. And if wherever we see yellow, means that the red axons are in close contact or in direct contact with the green axons. So we analyzed many different animals, many different embryos, many different stages. And every time we saw that these axons, the axons of the neurons that begin projecting from here, which is equivalent to this region here, they immediately go into contact with the green axon. So again, something similar to what we looked at what we found when we were looking at the migration of neurons. We said, well, we see the evidence, we see that they are interacting. Now we want to test whether that interaction is relevant for the normal stereotypical projection. So one way of doing this is, well, let's interrupt, let's interfere, let's mess with the green axon so that we alter the normal distribution or even the presence there so that we see how the red axon behave. And yeah, our prediction was correct. We use an animal, a transgenic animal, in which by expressing a toxin in using transgenic technology in the mouse, we can express a toxin on all the neurons that have that project these green axons. And that does not eliminate them completely, but it affects them dramatically in a way that the big pathways, the main pathways are altered, they are delayed, or some of them are completely absent. And what we see is that the axons of these neurons that normally project all the way from here, from the ventral midbrain, all the way into this striatum, now many of them stop projecting, and some of those that do project to different places, to incorrect places. Now, beginning from a basic observation of an interaction between neurons, now we have gone on the way to get the evidence that shows that interaction is important for their projection. So, and now putting all these types of evidence, this evidence coming from different laboratories and using approaches similar to this, using cultures, using knockout animals, using mouse, using chick, using rat, etc. We can begin to put together in a model to try to understand how a complex behavior of an axon that is born in this part of the midbrain projects all the way through the diencephalon, what I call the diencephalon, all the way to this striatum here. So some of the evidence that we have here in this summary is, for example, semaphorins, the role of the semaphorins, that's some of our results. This interaction between the green axons, now I switched the colors, but the dopaminergic axons are shown in green. This interaction between these green axons with the red axons, we know is important for this stereotypical projection. And we have put the evidence coming from other laboratories. This is not up to date because there are some other studies that have been continued. We have not continued this work. We have WINS, we have ephrins, we have netrins, slit molecules, etc. There are many molecules involved. And what we know now is that a complex behavior of an axon involves a complex regulation. Many molecules in time, not only in 3D, but in 4D, that is 3D plus time during development. So, and that gives us now a very good picture and people working on developing methods to let's say, to put stem cells in adults in this region to try to recreate the embryonic signals so that we can reproduce and put and connect this region, the midbrain, with the striatum in patients with Parkinson's disease. Now, there's a long stretch going from this basic information to a complex approach like that. But the basic information is this type of information that can be used for those approaches. Now, we go to a different perspective. We've looked at molecules, neurons, axons, and interaction between axons. Now we're going to look at how we can study brain development by analyzing the expression, not of one gene or a few genes, just all of the genes that are involved there. And it is a very ambitious approach, a very ambitious idea. And now with the next, what we call the next generation sequencing technology, it's a technology that allows the sequencing of a human genome in a couple of days. Now we can start to look at those things. And I mentioned at the beginning that by studying different species, different vertebrate species, we can learn about first about evolution, brain evolution, and also about how the brain develops in human. And in this experiment, we studied how we compared how the development of the telencephalon, which is this structure here, so the brain of this is a chick embryo, that's a mouse embryo. They are more or less at equivalent developmental stages, not identical because that doesn't really exist, but more or less regarding the brain, the stage of the development of the brain, they are at similar stages. So the telencephalon is here, what I mentioned before, that balloon that gives rise to cerebral hemisphere, and then the midbrain is here. In chick, the midbrain is huge, it grows a lot, and then the hindbrain would be along this side. And we have something similar in the mouse. It's a bit smaller, but the telencephalon is here, midbrain is here, hindbrain along this way. So, but if we look, let's say, if we look at the brain or even only at the telencephalon, it's just a tiny balloon in both species, in the embryos of both species. But how our interest, as I mentioned at the beginning, we're interested in how these changes in shape are regulated. These here at the bottom, these are models of a chick brain, adult brain, and that's a model of a mouse brain. The shapes, the size, the functions are very different, and they're coming from the same structure, from a very similar structure. So we decided to look at the early stage we could look at to analyze how, or rather, what groups of genes are expressed differentially that cause the divergence in the shape of this brain, so that their adult form is completely different, or at least very divergent. So this is the experiment we did. We took the telencephalon from the early stage. We could mechanically dissect it. So because there's one section beyond which, below which we cannot really extract it. So we obtained a brain from that stage, telencephalon from that stage, and telencephalon from this stage. And then we obtained from stage A and stage B, stage A and stage B for both, for chick and for mouse. So, and then we analyze which genes are expressed at this stage, and which genes are expressed at this stage, etc., for this one and for this one. And then we want to know what are the genes that are present in both animals. What are the genes that are upregulated, that is, are expressed higher at the second stage, and in both species, and then what are the genes that are expressed that are downregulated in both species. And we use here as a proxy for relevance it changes in expression. If a gene changes in expression, we infer that it has a relevant function in that process. And that's what we did. And some of the results, results are very complex, but I'm going to show you only some of the results. I'm showing them on the next slide. Well, this is just to give you, this is like a cross-section of a mouse brain and a cross-section of a chick brain. And what I want to mention here is that most of the genes that are expressed in one structure are present in the other one, in the other species. Most of them are the same, but the way they are expressed, the way they are combined, the level of expression is different. And so that is one of the main messages. So there is not that there is a gene network that gives rise to a mouse brain or a gene network that gives rise to a chick brain. It's rather the same network, but expressed or regulated differentially, right? Combined in different ways or expressed at different levels. And then one example of that is shown here on the next slide. This is just a very quick way of looking at things. What I have here is our genes that are upregulated or downregulated in mouse. Here on the left, these are genes that are upregulated in mouse or downregulated in mouse. It's in there, same for chick. Those that are upregulated in chick or downregulated in chick. And I put in red all the genes that are known to be involved in a function in the adult brain, right? And you can see that the genes that are upregulated, they all, or most of them, have been linked to specific functions in the adult brain. But remember that these early stages, you saw the embryos, there are no neurons, there are no axons, there's no glia, there's nothing that resembles anything in the adult brain. Yet, most of the genes that are upregulated are known for the function in the adult. That was a very surprising finding. So we don't know what they are doing. So we know that there are neurotransmitters, receptors, or multiple enzymes involved in the synthesis of neurotransmitters, neuromodulators, axon response, axon growth, etc. But we don't know what they are doing there. So their known function is in the adults, but at these early stages, no idea what they're doing. So there are many things to learn about these things. It's only that we need to look into it. And so this just shows one type of results that we obtained from this massive approach. We're looking at all the genes in contrast to what I described at the beginning, which is looking at a single gene. And then on the next slide, we see this is just kind of a summary of the results. We saw that most of those genes that we found to be upregulated or expressed highly in the brain of the mouse have been linked, these are some of the examples, have been linked to the autistic spectrum disorder, which is now considered a developmental alteration that leads to the spectrum, right? Which is a very complex thing to study. But the finding, it is very, very interesting that at these early stages, we're looking at the expression of genes that are known to be involved in behavioral alterations that are included in the spectrum of autistic spectrum. And that was a very interesting finding. So with this, this is the end of the brain development part. And just to remind you that we talked about the effect or the study of single molecules, neurons, axons, how they project, how they connect, how they interact. And then we looked at all the genes and how the expression of all the genes, studying the expression of all the genes, we can learn about how different structures diverge and how they are linked to the adult function of the structure we're looking at. Now, making a big shift, how we ended up looking at regeneration in lung gastropods, which is something we started doing recently. Actually, we started working on this from the beginning of the pandemic. It was difficult to work with animals in the vivarium, so we started looking at this. And we had the interest from a few years before that we wanted to know what was involved in the regeneration of these animals. this is going to be very brief. It's just like a I don't know, like a prime of the things we're looking at now. And then this is the type of animals we're looking at. These are land gastropods. You have seen them probably in your garden, and that's where we took them. We got them from our gardens to do some regeneration research. And the next slide, we see that why study regeneration? Regeneration has interested scientists for centuries. And we know that many structures in many animals are restored. Tissues, organs, body parts are restored. And so that the structures are restored and reintegrated into the pre-existing structures. Takes place in all animals. Some species, some of them have more the regeneration capacities more than others. And among invertebrates, only a few species have been studied in that. And so we started to study this one because it hasn't really been studied, and the mechanisms of regeneration in most are virtually unknown. So on the next slide, shows just one of those. There are several species we can study, and I show here. I put there all the structures that we know that this animal regenerates. Now we are interested in understanding what is the gene control, gene networks that regulate regeneration of these structures. And we have begun by studying the humble tail of the animal. And we decided to study this because we needed something simple to get started on, to develop the tools, the methods, the experience, so that if we understand what controls the regeneration of the tail, then we can go to study other complex structures. And that's how we went about starting this project. The next one of the things that indicates that these animals are capable of regeneration of every single tissue is an adult animal, if we starve this animal, the weight and the size of the animal will go down, and then below 40% of the adult size. And then if we feed it again, then it grows back again, indicating that all tissues can grow again. On the next slide. So we started doing the amputation of this cell, and then started looking at what are the genes that control that process. We don't have many results. In the next slide, we'll see. First, we look at the histology, the shape of what happens in the morphology, and we start to see the structures that form. On the next slide, we have started to do some characterization of this process. In this case, we're looking at cell division. These are in blue, we have cell nuclei, and in red, we see nuclei that are dividing, that are synthesizing DNA. So we can study cell division. On the next slide. And then now we can start to look at approaches similar to the one we use in chicken mice. We are going to extract this part of the tail of a regenerating animal and compare it to a tail in which no amputation was performed to see what genes are involved in the regeneration of this structure. On the next slide, then we can start again studying the expression of specific genes in specific times and places, and then we correlate that to the anatomy. There was nothing known about. I mean, of course, there's a lot known about the anatomy of these guys, but no systematic study had been done. So we started doing one, and we're building an atlas, an anatomical atlas of these animals. Now we can, whatever we observe in the gene expression studies or cell division, etc., we can put it in the context of the anatomy of this animal. And this is how we started looking at regeneration of this animal. I think it's the last slide. And this is just some things how, and just to recap all the things I have to say, is that we started studying brain development, looking at cells, at molecules, cells, axons, whole structures, And now we are moving into the study of regeneration using the same tools, the same approaches, the same basic knowledge, looking at development in the vertebrates, now applied to regeneration, trying to understand what are the basic mechanisms responsible for restoration of lost parts, lost tissues, which are present in all animals. And with that, I will finish and I'll have answer questions if you have time.
Questions that you might have? Yes? Well, I do have one. I'm not a biologist, I'm a theoretical physicist, but something that you have mentioned is this way in which the axon tries to reach the place where they need to connect. And one question that came to my mind is, for instance, how a specific axon knows that it needs to connect, let's say, in the muscle of my femur rather than this one. Because I am thinking that the muscles, they might produce the same chemical or something similar, indicating that I need a connection like this. And I don't know how they know that they need to connect in one muscle rather than
yeah, what very interesting question, and that's the interest of developmental biologists for a long time. It seems very complicated to understand going from one, let's say, from the spinal cord to a muscle in the foot, because it's a long way. But we have to remember that this takes place and at the earliest decisions are taken at the early stages in development, when the neuron is a few millimeters away from the foot muscle, right, or the leg or whatever. And there's a hierarchical organization of decisions. The first decision of the neuron would be to project, let's say, dorsal versus ventral out of the spinal cord. And then the one that projected ventral, then there's a second decision there that is to project to the leg primordium, what we call the leg bud at the embryonic stage. Then it already made it to the leg primordium. And then in the leg primordium, the leg bud, that will begin, the different muscles will develop. And then before the muscles are produced, you have a dorsal side of the leg primordium and then a ventral side. And there are some molecules similar to the ones I showed that are to which the axons are attracted and are repelled by. Okay, we'll go to the dorsal. Let's say that the decision was of a specific neuron type was to go to the dorsal, and the different neuron type went to the ventral side. Now let's focus on the dorsal side. The first decision was made. Now the dorsal muscles will develop, and then there will be additional signals that will allow the axon to go to muscle A and not to muscle B. So you can see that hierarchical decision system along time that makes a very complex decision, a very complex pathway that is based on not, I don't want to say simple, but simpler decisions. It goes along those ways.
Yeah.
And many things we don't know about.
Yeah. Questions, guys?
Yes, about the blockchain reporting. What do we know about this pathway? It's involved in other proteins, or how does it work?
Yeah, this